protein. So if protein is overexpressed, so then you can imagine they grow much faster, and that's the reason all these cells which are metabolically, including cancer cell, they have the overexpression of this survival. So one thing I want to emphasize here, I got a few grants. One of the two grants, I two PhD student, a potential PhD student which are very active or really dedicated for the research, and I also needed two postdoctoral research fellow for two different grants. So I'll introduce you a little bit from where I am. I'm from Australia. In Australia, I belong to Victoria. We work in Victoria. In Victoria, this state, we have three campuses in Victoria, one in Melbourne, another in Geelong. So 450 kilometers away from Melbourne, Warrnambool. So I work in Geelong. This center, this campus has a engineering, College of Engineering, Basic Science and Humanities, and nursing, as well as the School of Medicine. We got last 7 years. This is the School of Medicine. So we first batch events, and more than 30 seats for international students. So anyone who is interested can get admission this way. And these courses, they are really like US style. If you have a graduation, then you can enter in the teaching courses from 1 year, first year and second year, and for third year, different local in this region. So reason to develop this center, this School of Medicine, is to develop a strategy where you can these medical doctors in the regional area, because it's a very wide country, and the people or doctors, they don't want to stay or work in regional areas. So this school has been awarded to giving a boost to the people, or local people or regional people, so that retain doctors in regional areas. So these are the courses we have into medicine. We do the on degree PhD program in different courses in technology. We work on different aspects as infectious disease, immunology, cancer, or nanomedicine, or medical ethics and medical education courses in this center. We also recently last year started that's a optometry, and this course is very TH17, all these cytokine or these T helper cell, they play a role in cancer. So I have more than 17 students, including 4 postdocs, and they are distributed in different centers here in India. Our university have one campus in New Delhi with the TD, and one in Hyderabad. There are 4 or 5 students here in Hyderabad, and we have total 35 students in India as well. So I'm supervising more than 4 or 5 students in India, and we have more than 10 students in our center here in Deakin University. We working on nano delivery, starting from oncology, from neurobiology, cardiology, and different cancer, not only colon cancer. We work on skin cancer, liver cancer, and the ovarian cancer. So we have a various projects, and we have we are waiting for hiring 2 other postdoc and 2 other PhD students. So in the last 3 years, I got one grant. That's the grant is related to lactoferrin. That's the bovine lactoferrin and the buffalo lactoferrin. That grant we got from the Indo-Australian. We call here Indo-Australian, but in Australia we call that ASRF grant. And this grant was in collaboration with the PGH Chandigarh. So where they purify the proteins from buffalo milk, that's the buffalo lactoferrin, and the protein which we did in Australia, that's the bovine lactoferrin protein, and we share this reagent between each other. And in Australia, we did prostate cancer and colon cancer work with the nano delivery system, and here in PGI Chandigarh, they work on infectious diseases, including parasitic infections. So I'll go through today only the cancer work, but not cover the other one. In addition, I got another grant this year. That's on the LNA technology. I'll give you briefly what are the LNA technology, because you heard there are a lot of siRNA, microRNA, RNA. They are in our system, and they play a very high regulatory role to control any disease. So, but problem with this, all these, if you make an antisense or use these RNA, microRNA, the problem is they are very unstable. Their half-life is not more than 1 minute. So you can imagine how you can use these technology or these nucleotides to regulate the function and the structure and the function of these key molecules which play a major role in cancer and inflammation. So you need a technology. You need to stabilize these nucleotides from various nucleases, enzyme or other enzyme in our system. So this is the locked nucleic acid technology which we developed in our lab, and we found that there are few molecules which are first identified, the EpCAM molecule, which play a role for this for the spreading of the cancer and metastasis and invasion of all cancer types. So we make this molecule with the LNA. In addition, we also use these siRNA to survivin, which has been identified by 4 or 5 years ago, and make it LNA form. So we modify this as these siRNA and aptamers to EpCAM and make it a chimeric form in a combination, and we modify with the help of LNA technology to make it more stabilized. So this grant we got in collaboration with the Sankaran Netralia, with the Chennai, and the work which they will do, they will use these tools or these LNA technology chimeric form to kill the tumor cell as well as the cancer stem cell. What's happening when somebody taking a draft after some time, when they have in the stage 2 or stage 3, none of these chemotherapy is working. So be using these one, isolating the cancer stem cell from these patients, from retinoblastoma, from breast cancer and the colon cancer patient, and treating with this chimeric form of these nucleotides with the LNA modification. So I'll give you some brief over there. Before that, I'll give you a little bit brief which we got a success. This is called a translation research. In Auckland, we were working for a company that's a milk company, a dairy company, and that's the second largest cancer, that's a frontier, and their interest is to find out the nutraceutical, or as Professor Prakash had mentioned, the molecule which are naturally. So their interest is just to develop a nutraceutical from milk proteins or milk substance. So we purify a lot of components for them, but today I'm not touching all these molecules. I'll cover just lactoferrin. So we purify the lactoferrin from these milk from New Zealand milk, and we use this lactoferrin as a key molecule to treat the cancer. And it has been already identified that these lactoferrin, either from human lactoferrin or bovine lactoferrin, they have all these activities, starting from antioxidant, antiviral, or anti-cancer activity, and also help in iron absorption, because the presence of 14 to 15% of iron present in the pocket of these bovine lactoferrin. But we modify this protein, so we made this lactoferrin iron saturated, because this protein has 40% saturation. We made it 100% saturation, and we tested the different saturation stage of the level of this protein for anti-tumor activity and as antioxidant activities. So we found that if you use this protein as a 100% iron saturated, you will be able to cure the tumor in a synergistic way. If the tumor is small, if we start using these iron saturated lactoferrin oral diet, that you will not develop a tumor. So if we treat these mice at this stage, none of these mice means 10% mice, they decline in the tumor regression. But if you use a paclitaxel or doxorubicin, which are the routinely used drugs for this cancer, they have a very significant synergistic effect. And this synergistic effect is due to the presence of activating of the immune cell. So as the Professor Prakash mentioned, that immunity is very key play play a role for any disease. So if this lactoferrin, iron saturated lactoferrin, I'll give you more detail in the next slide, how this will evoke the immune system. So this evoked immune system, along with the chemotherapy, able to cure the tumor, and this we're doing in collaboration in some hospital in Australia and in New Zealand as well. So in my next talk, I'll cover about the mechanism over there. I'll summarize over here. So what happened, lactoferrin, when we give lactoferrin, iron saturated lactoferrin to these mice, the intestine has a lactoferrin receptor and also have the LRP receptor, and they play a role for the absorption of this protein and get protected from these proteolytic enzymes. Now question comes, this protein, iron saturated lactoferrin, why is it resistant to proteolytic enzyme? So we did in vitro and in vivo assay with the various cell-based assay with the cocktail proteolytic enzyme from gastric juice, and we have seen that if this protein is saturated 100%, either with the iron saturated or with selenium, none of these molecules for 24-hour digestion with cocktail enzyme get degraded. So this shows that this iron saturated lactoferrin somehow get protected from the proteolytic enzyme due to different structural configuration when you make it iron saturated, and go to the immune cells, and from immune cell go to the tumor cell where it start inducing apoptosis in these cells. So this is the interesting thing. This is the first functional medical ice cream food which we call recharge. If you are interested, just put in Google and find out about the recharge. So I'll tell you very briefly, this recharge ice cream with the help of the bovine lactoferrin, iron saturated lactoferrin, able to means have a synergistic effect or protect the intestinal or any side effect of any chemotherapy which people are taking orally in these patients, the breast cancer and the colon cancer. So interestingly, we are continuing this study, putting various saturation, different level of iron saturation with this protein, and working very nicely. So in addition, because dog, our pets, they are very well in Western country, they are very important for us. So we also using this iron saturated lactoferrin for dogs who have a cancer, and because in dogs, most of the dogs, means more than 70% dogs, they have a cancer after 4 or 5 years. So we are exploring the possibility for this further use. In addition, one of the drugs which this drug is already in the market, and this drug we investigated in 1999, and this is already in the market, and I think this also present in Sigma catalog. So this is DMXA. This has an antivascular activity. We developed this drug in Auckland University. So I'm not going into detail how this works and what is the structure of this one. Just to give you an idea how we are working towards the translation research. So now comes, when you talk about the proteins or siRNA or any RNA or any microRNA, how you protect these from proteolytic enzyme or from the nucleases, so from these systems. So important thing here, you need to protect your protein, peptide, and the siRNA so that they get absorbed from the system. From there, they get absorbed from these intestine. So, and how you can lower the toxicity? How you can control the cost of these important molecules? The iron saturated lactoferrin is very expensive. It's when you purify and make it iron saturated, it's become more and more expensive. So how you can protect? How you can deliver these molecule or expensive molecule to the site where you want to deliver. So we use a technology that's a nanomedicine technology or nano-based formulation, and we are interested more in, as I mentioned to you, the company is more interested in nutraceutical. They are not interested in any IV, IP or any injection. So we thought we spent minimum 1 year to develop a nanoparticle, and how this nanoparticle get protected from our acidic pH in the stomach and the alkaline pH in stomach, in intestine, and the different proteolytic enzyme. We already published this paper, so if somebody is interested, they can see in Nanomedicine. This is recently published. Another one we published in the International Journal of Nanotechnology, and these this already has been patented. If somebody is interested to explore the possibility to use any siRNA, microRNA, or any peptide to deliver it to the target site, just come and contact to us or put in Google Jagath Kanwar, you will see the patent details there and the application as well. So ideas here, we use a different polymers. So we use one core protein, the calcium phosphate, which give the retain the structure and the function of these proteins. Your calcium phosphate is a ceramic. It's a ceramic, it's hard, and it also binds positive and negative charge of these molecules. Besides that, we use a polymer which have a mucoadhesive capacity, because we want to deliver the molecule to the intestine, and from where it gets stick to the intestine, don't get diluted from various enzymes along with your food. So we want to retain on intestine, so that slowly it get absorbed from there. But how you protect these polymers from low pH in stomach? So we use another polymer to protect these encapsulated form of these nanoparticle with a special type of polymer, so that these polymer, these nanoparticles, they don't get degraded, and the pH, very low pH, even 1.2, don't play any very role. We did all characterization. We checked the pH stability, vary from 1 pH to even 8 pH, and we got the results over there. So this allow the sustained release of these molecule. And we not only tested iron saturated lactoferrin, we tested different proteins. We also tested microRNA, siRNA. I'll show you some of the data further. We did all characterization as everybody does, means about what's the size of the nanoparticle, how these nanoparticles go inside, and how these nanoparticles behave when you use a X-ray diffraction, and how they bind with the protein, how much they bind with the protein. I'll just focus you over here. If you see here, when we use these nanoparticle iron saturated lactoferrin protein nanoparticle and run on SDS base, you can see they stick over there. Those stick over there. And we thought we did something wrong. So when we follow up these nanoparticles and see the structure of the protein, run on Western blot, you can see these bands are still exist over there. So this Western blot shows that the structure of this protein is not altered with the help of this SDS space or these variable pH in SDS space. In addition, we also did microRNA. We check on different peptides, so they retain their structure and function with the help of these nanoparticle. So this is the data which we already published. This is in Drug Discovery Today. If you're interested, just figure how this will go over there. So these are the nanoparticles over here. They put a core there. They adhere to the mucoadhesive, due to mucoadhesive property, they retain over there, and from there get absorbed to the system. And it's not only through the endocytosis, or also through the exotosis, cytosis, and with this then go to the tumor site. From the tumor site, that has a LRP receptor, lactoferrin receptor, and some other molecule which through exocytosis go into the nucleus, and from where it release the drug. Here we mention lactoferrin, but they also release the siRNA or any drug here through the help of these different inside the endosome, and play a role for killing or disintegrating the nucleus, or whatever the action of your drug is there. So now we were interested because we were working on survival from the last 10, 12 years. So we were interested to find out what's happening when we feed mice with the iron saturated lactoferrin, and what the consequences in the mucosal immunity. So we took intestine from control and the treated by mice, and we do the microarray analysis. You can see a lot of protein, a lot of genes upregulated and downregulated. So we found more than 200 genes get regulated with the help of this iron saturated lactoferrin. But we screened some of the molecule, but interestingly we found the survivin have a major role. So what's happening when we feed these mice with iron saturated lactoferrin protein, survivin expression go down. So you can imagine the survivin is a protein which present in all tumors, and when it's overexpressed, tumor grow much faster. So when you regress this protein expression, you somehow control the growth of these tumor cells. So this is the structure of survival protein. This survival protein has been identified in 1997, and after that we start working on this iron saturated to making this dominant negative antigen or making an antisense of this gene, survival gene. We isolated this gene from mouse on the human, and we make it an antisense or a dominant negative gene, and we published a paper in JNCI and in 2001, and after that you can imagine more than 500 publications came out, and everybody is doing either regulation or these looking for the function of survivin protein with the different nutrient or with the different natural component or with the different drug effect. So, but recently there's one drug came out, and this drug in clinical phase 3 trial. Unfortunately, this drug has failed in clinical phase trial 3. But this shows that survivin overexpression, or if you regulate the expression of survivin, then you can regulate the tumor. So it's very, very important. Why this drug has been failed? Because this drug is derived from the pesticide, and you can imagine all pesticides, if you even modify or change the configuration of the or change the structure of these pesticides, they still have toxic effect. All the patients which they give this drug all have a vomit, high blood pressure, or also have a high in some patients vomit and the diarrhea as well. So this drug is failed in clinical phase 3 trial. So now it's very important. So we continue this work. So this is the survivin here. So this is survivin present in all tumor. So this paper has been published. This is in a review article. If somebody is interested, they can read more detail how this mechanism go, what's the different signaling molecule, how different signaling molecule play a role for this for the functional analysis of this survivin protein. In short, I'll tell you, this survivin is present on the spindle fiber. So when mitosis start occurring, the spindle fibration starts, and this is overexpressing over there. And this survivin help in cell division. So if you imagine, if you deplete this survivin, you can control the mitosis of the cell. This is a signal molecule. If you're interested, you can see the review article. So now we interested to make it a dominant negative the gene. And as I mentioned, we published this in JNCI, and all the tumor cell overexpress this survivin. But some cell, they are not a tumor cell, but they also have express survivin. And we found that these cells in nothing, that's an immune cell, that's an NK cell. And these NK cells have also start overexpressing survivin. So that's the reason when tumor is enlarged, survivin expression is more, immune cell also start expressing the survivin, and they're helping tumor cell to grow much, much faster. So our immune cell is behaving badly over here. So here we found first time that if the tumor is enlarged, this expression of survivin also go up. After that, a lot of paper has been found. So I'll give you small, when you use the gene therapy, either with the antisense or with the help of dominant negative protein, they will be able to regress the tumor, but they're not clear the tumor if the tumor is large. And they have very less effect if the tumor is very small. Reason, because at small tumor size, the expression of survivin is less. This technology or gene therapy is only applicable when the tumor is enlarged. If you use the combination therapy with the gene therapy with the co-stimulatory molecule, they totally regress the tumor. So now, gene therapy have a limitation because the GMOs, there are a lot of restriction to use the gene therapy. We were interested to use as a protein. But how you cargo this big molecule, 36 kilodalton protein to the inside the cell? Because all action of survivin happen inside the cytoplasm or mitochondria or on the nuclear level. So we use R9. This is a cargo molecule, cargo peptide. People use that. We use R9. This is a polyarginine molecule. So we make a complex, make a recombinant protein, purify this protein, and treated mice with this protein. So if you see here, when you use a combination with the paclitaxel or doxorubicin, they are able to regress the tumor, and not only one tumor, we tested different tumor types. So now, question comes because we have seen all these nanotechnology or nanoparticle which we make made intravenously. We give a route by oral route. This grow everywhere. They can't differentiate the site of the tumor. No doubt, at the tumor site there is more prevalence or occurrence of these nanoparticles, but they go everywhere. Now we develop another molecule in our lab. That's the aptamer to EpCAM, and this EpCAM molecule is overexpressed or always present on the apical side of the all cancer cell. This EpCAM also expressed in normal cell, but at that time this not present on the surface. So you can imagine if you have antibodies or aptamer which bind with specifically with this overexpressed EpCAM antibody, if you tag with your nanoparticle with these aptamers, then you can deliver or target the tumor site. So we did all these studies. We did the tumor study internalization. More than 90% cells internalize these nanoparticles when we use the EpCAM tagged aptamer. We did the all NLC functional study, how this nano delivery system with oral delivery with this technology, how able to regress or stop the function and the function of this survivin protein. If you see here, when we use this survivin mutant either with the siRNA or with the chimeric form, they totally regress the expression of the survivin. There is no expression of the survivin. When you use the control, you can see at different stages of mitosis, you can see the expression of this protein in these cells. Then we check the stability of these nanoparticles with the different pH and check the internalization with the flow cytometer, how much cell get transfected or take these nanoparticles. So most of the cell, more than 80% cell, in some cases, some tumor cell, colon cancer cell, stem cell, in CACO2 cell, they take more than 90% of the cell. Reason CACO2 cell is a differentiated cell line, and they have a more structurally which have the capacity to engulf these nanoparticles. So this is the another study which we did where we use the CACO2 cell line, use these siRNA, and these siRNA with different dilution, they're able to completely kill these tumor cells. So then we did the siRNA, did the nano formulated form of this siRNA with the help of another microRNA which you use the RNA21A antisense. And this microRNA, when we use in a nanoparticle, they're able to regress the tumor or completely regress the tumor when we use in combination. We tested not only one breast cancer cell line, we tested two different breast cancer cell lines, and the cell line which is colon cancer cell line and the lung cancer cell line. And we also see the action of this nanoparticle, these special type of nanoparticle with the siRNA, whether they are mediated by the EGFR positive or EGFR negative cell line. We tested the two cell line, which one is positive and one is negative for EGFR. So these nanoparticles, they don't need any receptor, they don't need any mechanism. They go through endocytosis or through exocytosis. So this is here we feed these mice with these nanoparticles, cut this section of these intestine of these mice, and you can see here, this is with the rhodomain, see the nanoparticle, they're going inside through the intestine. So we did some functional assay. So when you use these survivin antagonists and how they play a role on regulating these molecules which have the function or for invasion, metastasis or growth of these tumor cell. You can see over here when you use in comparison with the drug. So our technology with the mice, microRNA, siRNA, nano medicine-based nanoparticle with oral delivery, able to regress more function or expression of these important molecule for cancer therapy. So this is the key figure which we already published. Yeah. So here this is the molecule which able to target these points for the protein level. So now we are interested in multifunction nanoparticle, to use a nanoparticle as a vehicle to transport the multiple targeted molecules or multiple drugs to the site of tumor. So here we develop, as I mentioned, in our lab we develop APCAM molecule, and these APCAM molecule we develop with the help of this method. We made a LNA. LNA is locked so that these nucleotides they are not prone to different these nucleases. So here we make it a cyclase. So this is the molecule. If you are interested, you can read more detail about the LNA technology in our recent publications. So here we making either chimeric form of these aptamers, LNA-based aptamer. We making with not only one lactoferrin protein. We making with siRNA. We also make using these microRNA or using different two types of aptamers. Another aptamer which we use the nucleolin aptamer, and this aptamer has the tendency to translocate protein, these nucleotide from outside to inside. So and this there are I think more than 15 publications has already been published, and this is through antisoma. So what we did a new one. We modified this one because the stability of this the antisoma is using. They're using these aptamer to nucleolin, but their nucleolin aptamer is not locked or not LNA. We use the LNA one, so we increase the stability. So you can imagine if you put all these molecules on top of these nanoparticles, making it a multi-functional one, you can target more specifically to the tumor site. You will get answer in this slide. So answer here, when we use different colon cancer and breast cancer, and when we compare with the normal intestinal cell we used here, you can see over here, normal cell, they don't have toxicity. However, the other breast cancer cell or the colon cancer cell, they have a high toxicity. So means they internalize more number of nanoparticle than be tagged with multifunction LNA tagged these aptamer, nucleolin and these aptamer, then the aptamer. Then we see the biodistribution assay, whether these nanoparticles, they go more specifically to tumor site or going everywhere. No. Answer we got, when we use a nanoparticle along with our nuclear line with the points one, you can see here they go everywhere. Black dotted line, they go everywhere. Although they're going more towards the tumor site, but it's going everywhere. So when we use LNA or a combination with the LNA technology modified nucleolin and APCAM, you can see here they go more specifically to the tumor site. They are not presenting on all way. Intestine, we got intestine, but you can imagine the nanoparticle which we make, we have seen if you feed once, even after 7 days, you can see these nanoparticle in their stools, in their intestine. So they stick because it's a mucodisc property. But we don't mind, because if somebody have a colon cancer, and if you use these nanoparticle for delivery, any drug, they will be able to deliver more specifically to the cancer or the colon region. So here, if you focus here, these tumors with the breast cancer and the colon cancer, they more specifically go to the tumor site. So we got the answer here. If you use the multifunction nanoparticle with the LNA technology based these nanoparticle with nucleolin and APCAM aptamer, you more specifically target the tumor site. So we are going towards more sophisticated technology where we are using these multifunction nanoparticle cargo with the Cy5Y, some the chromogen, or using an oxygen sensitive biosensor, so that we can test and monitor the drug effect of this drug in a different scenario. So you just imagine over here, if you have a drug, you put your peptide, siRNA or any drug here, and use these nanoparticle with the help of oxygen sensor and tagged with the LNA modified aptamer, and feed these mice, and you use these one for imaging as well as different other treatment. Here we are using a cell line from the patient, which are CD33 and CD44, 133, CD36 positive cell. We sorted the cell, injected into the xenograft model, and from xenograft model, we treating these mice for these colon and the breast cancer. So the technology which we develop over here is just to summarize here. So technology technology we develop here, that's the stem cell treatment and cancer cell as well. So if you have a cell which are positive for these markers, so means these cells, they are stem cell. And now we are expanding this technology. The grant which I recently got from NHMRC, from federal government of Australia, we are using these sorting these stem cell from the patient who are not responding to any drug. So ladies or who have a breast cancer and they're not responding to any drug, so we're taking those tumors, separating these tumor cells, and those cells, we label them cancer-resistant stem cell. So we're using these cancer-resistant stem cell, developing in tumor, and start treating these mice with the help of this technology, nano delivery system. In addition, initially we also use this therapy. This is the photothermal therapy and the magnetic therapy, because the nanoparticle which we have with the iron saturated one, they have a magnetic property as well. So we use magnetic touch on the tumor side along with this one. So with this one you can use, and we tested this in mice. Even after 15 days you can do the MRI, you can see the effect or monitor the effect of your drug in these mice. This is my, I think, last slide. So in addition, we also develop a nano capsule, and this capsule is not bigger than a capsule. It's a very small nano capsule, and in our engineering department we are again standardizing this nano capsule for different diseases. So one idea here which we tested in a rat model, we didn't get success. I think we did 5 times in different model. Mice and rat, we didn't get success because this nano capsule form, they don't go with the intestine. What's happening when there is a food or not a good peristaltic movement in the intestine, these nano capsule or micro capsule, they don't pass through the one. Even we have a control over here. We can control the movement of these nano capsule. But problem is this: if we give food to these mice and give this micro capsule, then they don't go properly, they don't behave, or we don't have much control on these. We did all study. We increased the efficacy, we increased the voltage, we increased so many things. We didn't get a result. So what we have to do, for example, somebody have a problem, means infection in the intestine or have a colon cancer, then we have to put these patients for 24 hours on fasting. That's 100% sure. So if we put these patient on fasting and give a gel to these we did this on an animal study, gel in a viscous material, then you put your these micro capsule, then you can control, working so nicely. We tested these one, and this technology we already patented. So this what's happening, we testing this one on different inflammatory diseases. As Professor Prakash mentioned, IBD is a major problem in Western country. IBD not in Western country, here in India as well. So we want to test this technology for IBD, colon cancer, parasitic infection, bacterial infection, any mucosal immunity. Good thing with this nano capsule is this has a different chambers, so you can put different molecule, different drugs. So in combination you can combine. And good thing with this targeted one, you can control the movement of these micro capsules. So from the computer, from here, you can imagine if somebody have a parasitic infection and you use this micro capsule and deliver your drug to the site where you want to deliver, you can control the toxicity, you can control the dose of these expensive drugs. So from this I stop my talk. And these are the key people who are responsible. Dr. Rakesh Sahgal from PGI, we got this grant in collaboration with him, and we did a lot of study in PGI Chandigarh, parasitic and bacterial infection study in rat model, and the funding which I got from private funding from Frontera, from WBC from Australia. This is again from Australia. This is got from osteoarthritis. The other interest is osteoarthritis. We got from funding private from Vision Australia as well, and we got funding from an HMRC from Australia, government funded. This is from Australia. This is again from Australian government. This is Indo-Australian funding from our university and from Auckland. So these are the people, and these are the different institutes here in India and in New Zealand and Australia. We are working in collaboration with all these people. So thank you very much. These are the people. Any question?
a very huge amount of work, a good piece of work. I have 3 questions. One, lactoferrin. We assume that in a lactating woman, the amount of lactoferrin will be high. So do you think they are not susceptible to the breast cancer?
They are not susceptible to breast cancer?
Breast cancer, because lactoferrin amount will be more in the lactating woman. Second question is, with age and someone develops lactose tolerance, so what will happen to those if you give orally the saturated lactoferrin? And third, the cancerous cell takes the strategy to throw out the particles by using proton pump. So what are your comments on these 3 questions?
Yeah, very good questions. I love. Yeah. So first question is how, or ladies who have a more human form of lactoferrin, if they have a cancer, how this therapy, our iron saturated bovine lactoferrin play a role over there. I'll tell you, we tested these therapy initially, dose, how much dose we need to do and how much the toxicity. If you talk about the iron toxicity of these, when we feed these one, we use 1% of diet. 1%. And if you compare the supplement of iron, which we take routinely, is, I think, just 25%, even just less than 25%. So toxicity of iron toxicity is ignored. So that's a different story. I'll talk in next talk how iron saturated lactoferrin is good for RBCs, how it's good for ladies who have a iron deficiency or some toxicity problem or have a breast cancer. For breast cancer, the human lactoferrin have a different mechanism for controlling the tumor, and the bovine have a different mechanism. And the mechanism, the lady, we already have a human form of lactoferrin. In their case, we feel survivin expression is regulated, so there is no much effect on regulation of these of survivin. So when we feed these one, we haven't done any clinical trial for breast cancer, but my assumption is when we use iron saturated lactoferrin, it go in a different way and activate the immune cell. I didn't put this slide. Next talk I'll show you how this awoke the immune system. So as for the toxicity concern, this protein is, we are taking milk from so many years, and this lactoferrin already in the market, and the people are taking 600, recommended dose is 2 capsules, means 300 milligram per day. So if you compare those things, the other dose is quite low. So one question is, I don't think it's a toxic, and we did all inflammatory study and feed mice for more than 90 days, and we didn't find any side effect of this thing. In vitro, we tested different cell lines, normal breast cancer cell, human one as well, tested different doses. We didn't find toxicity. We got a little bit toxicity when we go 64,000. But that toxicity is not related to breast cancer only, that also with the normal cell. But that mechanism is totally different. Because this is a big molecule. You just imagine it's a 80 kD molecule. And you're putting 34 or 64 milligram protein in a patty dish, that'll create a hypoxia. And that another mechanism. So that mechanism is totally different from this mechanism. And we found that when we give this lactoferrin, the lactoferrin itself internalize into the inside the cell. Within 1 hour, it will cross through the membrane. So it will go to the nucleoside and the mitochondria and depolarize these mitochondria. So when depolarize the mitochondria, it suppresses the expression. You can imagine because survivin is also present in mitochondria. And mitochondria is the powerhouse for controlling all energy sources. So it controls over there. So this protein has a multifunction. That's why I put it here. It's a multifunction protein. And somehow it's playing a different role in dose-dependent manner, and in other cases, it's a different role. And your second question is?
Lactose tolerance.
Yeah, another good question. The lactoferrin, this protein is totally different from lactase deficiency. This is a lactoferrin, where that is a different thing. And there is no any side effect and no any toxicity for this reason. And this is already in market. People are using in USA or in a different country. So there is no toxicity. And people are
taking toxicity. What I'm trying to know.
Tolerance, yeah.
Yeah, lactose tolerance. What happens? The patient who develops lactose tolerance, nothing from the milk they can consume inside the body. Rather, it is a deleterious effect.
But the protein which we are talking about, that's a 100% purified lactoferrin. So it's not lactase or some other molecule attached with this one. There is a question for LPS. People say it has some LPS or lipopolysaccharide attached with the lactoferrin, purified lactoferrin. But the protein which we using over here, we depleted all these LPS effect with the different technology we develop in lab. So lactase is a different and lactoferrin is a totally different. We don't have any cytotoxic effect.
So my question is, we are preparing nanoparticles, sir. So stabilization of nanoparticle is a major issue. Basically, you are taking proteinaceous drugs. Protein shows instabilities. So how you are stabilizing? And as you are taking sodium alginate and chitosan is a carrier, so they are excellent mucoadhesive properties. They can adhere throughout the bodies. But your target is colon cancers. So instead of that sodium alginate and chitosans, we can try for other carrier also. Those are having excellent mucoadhesive properties basically in the colon. And what should be the effect of combination of chitosan along with sodium alginate?
Yep.
Please explain.
Yeah, we tested this initially in cell line normal colon cancer and the CACOS2 cells and FHS, and that's a normal intestinal cell. We tested all doses and we standardized. You are absolutely right. If you use alginate high dose, that itself killing the cells. Mechanism we don't know. There are a lot of mechanism but we standardized the dose in such a way that when we feed these mice, including 1 gram of in diet, they have that much concentration. And we use a void nanoparticle in a control. Our nanoparticle in void, when we feed these mice, they don't have any toxicity or any side effect in it.
How you are optimizing the formulation?
Formulation we optimize, as I told you, first we see the stabilization. If our molecule is not stabilized with the nanoparticle, there is no question to feed mice. Yeah? So we did the stabilization with different temperature, different pH, and different humidity control. Because that's another question. If humidity is high and you prepare your nanoparticle, keep in a fridge at 4 degrees centigrade, all coagulate. So we use secretly another polymer, another thing, with that retain the structure of the nanoparticle. And when we use that molecule on top of these nanoparticle, that shrink the nanoparticle a little bit.
Doctor, that's that is the shape or the size of the nanoparticle.
Yeah, size of the nanoparticle is vary from 200 to 250 nanometer. So that's the range we got. We work very hard. I tell you, we spent minimum 1 year just making the right size or the right nanoparticle for our study. We initially got the microgram, 800, 500. We spent a lot of time spinning, different temperature, different speed, so many things. But at the end, we found this is the last we can go. If we go lower than that, our protein, they don't bind properly, or we don't get equally spherical structure of our nanoparticles. So we standardize that if you go for 180 to 230, then that's the last resort. And that's not only for one protein. We tested different protein with different microRNA as well. So you just imagine if you have a nanoparticle, and for stability, in addition, we also did the stability. You can imagine transporting material from Australia to India in summer, temperature in a flight, how much it go up. And we tested this one, different batch, 6 months and 1 year after 1 year. So after 1 year, same effect. We didn't find any side effect. We didn't test on tumor one, but we tested in parasitic infection. We didn't find any effect after 1 year when we make these nanoparticles. But controlling these size and storage of these nanoparticles is very, very important.
Thank you.
Great job. I had a question regarding the lactoferrin receptors you said are found on the tumor cells, a variety of tumor cells that you tested, but also they are found on immune cells. So on the one hand, you're saying that this induces survivin depletion, and that's the reason the tumor cells become susceptible to lysis. Whereas on the other hand, the immune cells also will undergo immunosuppression will occur. So how do you differentiate, or what?
Exact mechanism, we don't know exact mechanism, but we did all apoptotic markers. We tested all immune cells, CD4 starting TH1, TH2, all immune cell. If you use lactoferrin very high dose, then they start inducing the cell death. But you just imagine this is a very big protein. But at lower dose, which we feed mice with 1% gram, they're not playing inside. Another question is how is to transport this one. We use the labeling of our nanoparticle. We also label the immune cell and put these one in intravenously in different routes, and we found they able to cargo and protect. I'll tell you, if iron saturated lactoferrin that protect the immune cell mechanism, we don't know how this is working, but this protect the immune cell. And in next talk I'll tell you how this work. It'll happen through peer patches. So we tested through peer patches by labeling these nanoparticle our protein as well, and we found same protein in a tumor cell. So we assume that this is going through the immune cell somehow, and when immune cell going to the tumor side, able to kill the tumor cell or present these cells in such a way that they induce the cell death in the cell. Yeah, but one question I forgot to answer your question. Very interesting question. How it affects on the PGP? These are the drug resistance molecule. And drug resistance, these PGP play a role for taking any drug from inside to outside. So this is and all cancer cells have, don't deny, all cancer cells have high expression of PGP. This is drug resistance protein. We tested this one. Good thing with our other lactoferrin, if we use lactoferrin, it's down-regulate the expression of the PGP protein as well. So you can imagine a protein, and mechanism is not only through PGP. Other nanoparticle have a different role. That's the endocytosis, exocytosis, or transcytosis mechanism. So when nanoparticle go inside, we assume, and we did some in vitro assay in our study with the 4 or 5 different cell line. When they release these nanoparticles, after 48 hours, the expression of PGP go down. So you can imagine, if somebody have a drug resistance problem, and if you use a lactoferrin, I assume this may facilitate or help in those patients as well. So, sorry, I forgot to answer this question. That's it.